Hello, everyone. My name is Joe Hacia. I'm a geneticist on the faculty of the Keck School of Medicine of the University of Southern California. And today, I'm going to give you a brief overview of mouse models for adult Refsum disease research. As you may know, DNA is the instruction book for all life. And if you were to look at a cell from a mammal, you would find in their nucleus that it contains DNA. It wouldn't matter whether that cell came from a mouse or a human being. So we often use the term genome when we think about DNA. And the genome is the entire set of DNA instructions found in a cell. In humans, the genome consists of 23 pairs of chromosomes located in the cell's nucleus, as well as a small chromosome in the cell's mitochondria. This is not to be confused with a genome, which is my favorite Halloween costume. Here are some fun facts about the human genome. It's about 3.2 billion letters long and comprised of A, C, G, and T, which is shorthand for adenine, cytosine, guanine, and thymine. It contains about 20,000 genes that encode proteins. And if printed out on paper, the entirety of the human genome, that stack of paper, would be the size of the Washington Monument. So every cell in your body with a nucleus contains this amount of information. Now, this might beg a question. If you were to look in a crowd of human beings, you might ask, how similar are everyone's genomes? In this case, one would expect them to be about 99.9% the same. Human beings have very similar genomes from one another. So really the focus of this talk has to deal with some disease-causing misspellings in a person's DNA. And it's important to note that all of us have disease-causing misspellings in about 16 to 20 genes responsible for recessive genetic disorders. And most of the time, the backup copy of the gene from mom or dad is functional, so the person is healthy. And in people with adult Refsum disease, most typically, they will have misspellings in both copies of the Phi-H gene, one that they inherited from their mom and one that they inherited from their dad. So let's just kind of put the terms gene and DNA into context. There is a concept called the central dogma of molecular biology, where we think of DNA or the genome in a person's cell as being like the World Wide Web. It contains the instructions for the entirety of the body plan of the human body, going from development to the adult state. Now, DNA itself can be transcribed into a molecule called messenger RNA. Now, what is that? Well, it's very similar to thinking about a web page on the World Wide Web that gives you instructions about how to build something. Let's just give an example about how to build a car. Now, this messenger RNA in and of itself will then be translated into a protein. And that's similar to using the information on a web page to manufacture something like a car. So let's give you an example of the human Phi-H gene sequence. Here it is in all its glory with its A's, C's, G's, and T's. And what I'm going to highlight in yellow are the regions of the gene that give the instructions for encoding the protein. Okay, so you see it's only a small amount of that entirety of that sequence. Now, since the focus of this talk is two genomes, mouse and human, let's talk about a tale of the two genomes. Similar to the human genome, the mouse genome has about 2.5 billion letters, A's, C's, G's, and T's. It also contains 22,000 genes that encode proteins. The mouse, in and of itself, the genome, also has a Phi-H sequence. Here it is in all its mousy glory. And if one were to highlight the parts of the gene that give the instructions for making a protein in yellow, you would see it on this particular slide. So if you were to then compare the human and mouse Phi-H proteins and look at, a predicted structure you would see that the structures of these proteins look like they're separated by birth. They look very, very similar to one another, the panel on the left and the panel on the right. Both are 338 amino acids in length and share over 78% identity. So the only conclusion one could come up with from this data is that, geez whiz, we aren't all that different after all. Just a little pause for a little chuckle, hopefully. Now, people and mice also have many similar physiological features. When you think about this, a human and a mouse have a brain, a liver, a lung, muscles, and so on. And in fact, some mice are arguably even more similar to humans than others. So it is possible to genetically engineer mice to have mutations in any gene of interest. And in fact, this has been done by two of the Global Dare board members, Sasha Fernandez and Ron Wanders. And this was a scientific manuscript published in 2008. And they found that mice lacking a functional copies of the Phi-H gene develop a condition like adult Refsum disease if exposed to dietary phytanic acid. And in fact, the phytanic acid levels are elevated in their tissues if exposed to phytanic acid-enriched diet. So if you look at the panel on the left, on the y-axis, you'll see the amount of phytanic acid levels in nanomoles per mg of protein. And if you were to look on the x-axis, on the lower part, you'll see animals that are on low, medium, and high phytanic acid diet. And you will see animals that are controls in ARD, which is adult Refsum disease. So the animals with adult Refsum disease that are on medium and high phytanic acid diets have elevated levels of phytanic acid in these various organs. All right, so just like you would expect from a human being. And in fact, there is evidence of ataxia and a peripheral neuropathy in these animals as well. So what can we do with that? Well, one can use the mouse model to try to understand why phytanic acid is toxic to the body so that we can figure out how to prevent and stop tissue damage. In addition, it is possible to test the safety and efficacy of therapeutic interventions in mice, such as gene therapy, to improve phytanic acid metabolism, or drug therapies to improve phytanic acid metabolism. So I like to leave on a quote from a famous Western philosopher, a very wise human being. I only hope that we never lose sight of one thing, that it was all started by a mouse, from Walt Disney. Thank you so much for your attention.